Okay, good afternoon. Welcome to the Asia Symposium, as we call it. First of all, I would like to thank all the sponsors of the conference, all the organizers, and obviously the participants and the presenters. And this is basically to bring together all the people who are either from Asia or working in Asia for biodiversity informatics in general. So that's the purpose of the symposium, because we realize that there are not many teams working, and this is an opportunity for us to get to know each other and see what everyone is doing so that we can build more collaborations. So that's the purpose. And I don't want to take more time talking about anything and just invite the first presenter to start the session.
Hello everyone, I'm Aino from National Museum and Nature Science, and I will be giving a presentation titled Importance of Language Localization, Attempts for Collecting and Publishing Specimen Data in Japan. In the field of science, English has become the de facto standard for resource sharing. However, for us in Asia, English can often feel less accessible than our native language. Simply translating words is often not enough to fully capture the meaning, as cultural background and community needs must also be considered. This is where language localization comes into play, adjusting and reflecting the language to fit the needs of specific linguistic communications. Therefore, to promote effective utilization, we need an ecosystem where everything can be completed in our native language. The members of JBIF are engaged in language localization for JBIF, resource on biodiversity information, and we are also promoting their use within Japan. The role of JBIF is carry out active relevant to JBIF in Japan. Its main responsibility is to promote use and dissemination of biodiversity-related data. We are creating an environment that is user-friendly for non-native speakers. Let me introduce the activities we are undertaking to achieve this. We are providing resources for collecting and utilizing biodiversity information on Japanese. We conduct workshops and lectures on biodiversity information. We also handle translation of the JBIF website and science review. Science review is also available in print. If you'd like to take a look, just let me know. And we make some pamphlets. One of our key initiatives is the mass management of the SNET portal site, where we focus significant efforts. Especially, SNET is one of the highlights of our activities. The SNET site is managed by the National Museum of Nature and Science. SNET collects natural history specimen data from museums and research institutes in Japan, ranging from small to large scale, and publish this data in GB through the SNET platform. Using SNET, we also disseminate biodiversity information to the international community to GBs. Additionally, we provide a Japanese language search portal for domestic needs. The input format for SNet is designed to align with the GB occurrence squad terms. The input fields are configured in Japanese and include unique fields for domestic use, allowing use to input data entirely in Japanese. I'll zoom it because it might be small. As you can see, all the fields are set up in Japanese. So even if you don't speak English, you can still input data and the data probably published GB. Not only the system, but also SNet administrative data entry support is conducted entirely in Japanese. This is my main mission. We create and distribute introduction manuals, perform human checks on data, and provide help desk support all in English. All texts are carried out exclusively in Japanese. Since it's created by people who speak Japanese, it's naturally in Japanese. Within this approach, we have collected a large amount of data from small-scale museums as well. Since the SNet launched in 2005, we have entirely 77.3 million data records in approximately 20 years. I'm sorry, in Japanese, this chart is blue line is open the data, 7.3 million, and red line is institutions, about 120 institutions published that data. So why have we actively addressed and organized on Japanese name to this extent? It's largely due to the specific name of Japanese name used in Japan. What are Japanese name? Japanese name called wamei. They are the name of organisms commonly used within Japan. There are mainly 4 types. But as shown in this diagram, there are overlapping areas and varying difference among different people. Explaining all of those work take up the entire presentation time. So today, we will particularly focus on Japanese standard name based on taxonomic backgrounds. Wamei is a kind of name for an organism written. Wamei is kind of name of an organism written by Japanese. Critically, many taxonomic groups have Japanese names that correspond to each taxonomic concept one-to-one. Because of this, more popular and frequently used than scientific name in Japan. Therefore, it's essential to support searching using Japanese names. However, not all species have a complete list of Japanese names, and some species have overlapping local names, making things quite complex. Thus, a search system that includes wamei is essential. This highlights the need for specimen checklist. This covers wamei names. You are all familiar with this label. Specimen label is the primary source of occurrence data such as scientific name, date, and locality information, which are difficult to digitize for global use. Handwritten note and Japanese error dates add complexity. For example, this label is it's unclear whether the date on this label is in Western or Japanese calendar format, or if it's April 20th or December 4th. There is a risk of incorrect data entry due to misinterpretation by the interpreter and issues within handwriting. This can lead to the loss of information through digitization. For example, handwritten labels, sketches, illustrations, etc. In addition, there is also the risk of typing errors and misinterpretation. Publishing label image offers the advantage of making the information more accessible and allows for direct access to primary data. Digital data often becomes secondary information filtered through the data entry person. Label image and ensure data verification, so it is necessary to make label image available for public access. Digital archive is the current trend in digital image achieved in Japan. The images of specimens, including labels, which I introduced earlier, the Japanese government requested all museums to publish images online. Japan has cross-search portal site called Japan Search for various resources are available. SNet provides occurrence data to Japan Search, but image data support is limited. For smaller museums, creating systems for published images externally can be challenging. Therefore, it is hoped that SNet will also assist with image publication. There are expectations for SNet to offer further support, such as providing how-to guides and systems for image publishing, and to promote the sharing of specimen images, include lab images. We have made significant progress in language localization, but there is still room for improvement. Additionally, we share specimen information properly, both within the country and globally. We plan to develop national checklists for backbone taxonomy, including their common names called WAMEI. We want to provide a search service with WAMEI for entire GBIF data by converting WAMEI to specific name. And we want to publish image data to ensure data verification and to address domestic needs. If those goals are achieved, we expect data collection and publication to become even more active. If our efforts provide highly effective, we believe our activities will serve as a model for others. By sharing this effort, we can connect Asian collaboration with the global research community, building bridge between Asia communities and the global research community. We will also promote collaboration between citizens and governments, further enhancing their cooperation. While we believe our activities will serve as a model, we also think there are better approaches. Let's actively exchange information within Asia. Thank you.
Thank you. I think we have one quick question if anyone has, otherwise we should have some time at the end to discuss more. Okay, we can have the next presentation. Terry, please. Thank you so much.
Okay, thanks. Thanks, Vijay. So my name is Terry and I'm working at Paris Herbarium. I'm currently a biogeographer and project manager in Biodiversity Informatics, but today I'm going to talk about the role of tools and technologies in enhancing human knowledge acquisition. And I will do that taking the floor of Cambodia as a pilot project. So how did it start? Last year, I gave a talk entitled "Do Our Project Delimitations Display a Continued Legacy of Colonialism Towards an Independent Flow of Cambodia?" Because at that time, I was thinking about this question of decolonizing our collections, particularly because Cambodia was part of French Indochina colony since 1862 until 1954. And this long presence of France in Cambodia produced the biggest collections of Cambodian plants hosted in Paris Herbarium. The flora of the region started with, with the Flore Générale de l'Indochine in 1907, and it has then been published as the Flore du Cambodge, Laos, Vietnam, when the independence have been given to the countries in 1960, and it has been published by the museum in French only. It is now jointly published with the Royal Botanical Garden of Edinburgh. since 2014, and it's published as the Flora of Cambodia, Laos, and Vietnam in English only. So the question of providing a Flora of Cambodia is also to give botanical content in Khmer. So how to construct the flora? As I said, I'm not a botanist, so I just started to sketch, on one hand, the resources that were available at that time. So the collection, I tried to organize that with different items, collections, publications, and also the old collection that we have in Paris Herbarium. On the other hand, I have listed the open tools that are currently available to collect and sort out data and link the data as the online databases, for example. And so the first things I've done to create this flora, oh no, sorry, that's another thing. I ended up with this sketch, which is certainly not totally exhaustive, but give you a kind of a start to really construct the flora of Cambodia, and as a first output, a checklist of Cambodian vascular plants. So the first thing that I've done is that we had an open call from a citizen science program at the museum, which is called Les Herbonautes, which is used to transcribe the labels of the specimens. And because of this long presence of France in Indochina, in Cambodia and Indochina, we have a lot of specimens labeled Indochina without knowing where they come from, really. So that was the first things that we've done. So we applied for this call, and this mission, Flore du Cambodge, has been set up, and it's now finished. So we have 4,600 specimens that, with all the metadata collected, and unfortunately, I cannot show you the result for now because I'm still waiting for their team to provide it to me. But that's a very good point as a start, and it will not only help flora of Cambodia, but it will also help the flora of Vietnam and the flora of Laos. The second thing I looked at is the publication, especially this one, which has been published 2 years ago, the checklist of the vascular flora of the Sunda Saul Convergence Zone. And I have extracted from this checklist the names of the Cambodian plants because I wanted to match that list to World Flora Online backbone. So I got a little less than 2,300 names. And with the help of Alan Elliott, the 10 manager of World Flora Online, we had these results, 88% of positive match, which is rather good. And so we have now a list of 2,020 names with World Flora Online ID. And what is very interesting with this tool from World Flora Online is that when the match is not positive, you have candidates that are highlighted. So that means I will have to go into more checks for that. The third tool I would like to talk about is Echinopsis, which is an extensive notebook for open science on specimen, and it's currently developed by Nikki Nicholson, Eve Lucas, and Isabelle Laredon at Kew. It allows you to access to different collections, to take notes, and to keep track of the data. And I believe there is a demo tomorrow, and I see that Nikki is here, so she probably can confirm that. So this is how it looks like when you install the tool on your personal computer. This is your vault, and it's very simple. You can access to these different databases by just using this command panel. So you can access to IPNI, you can access to GBIF, Bionomia, Geocycle, and also Crossref for the literature. So I won't go into all the vaults that I have created, but I have just highlighted some of the points to show you how easy it is to use it and how useful it is. So for example, through Geocycle, I have selected all the herbarium that have currently Cambodian specimens. And this is an example of the herbarium at the Royal University of Phnom Penh. And you can see that you have different properties. You can you can sort out your data, you can add tags, and also you have on the back the local notes where you can also add any information you'd like. This is an example for IPNI. So from the checklist that I talked about earlier, I have sought to integrate the data that are present in Cambodia. And this is, for example, Melanolepsis vitifera. In the locate notes, I have here highlighted a link to a specimen that we have in the museum. And as you can see, this specimen is a holotype. So in my vault, I have added a tag here, holotype, and that would help me to find them out if I want to see them later. So yeah, the link to the checklists, and here is just to show you that the checklist has been also retrieved from Crossref, and he's here in my vault as well. So I can link literature to the specimens and to the names. There is a functionality with maps, so you can highlight those specimens on the maps. And for example, here in green, it's a type specimen that I have retrieved from GeoBIF occurrences. And in red, I have highlighted the type specimen that we have actually at the museum, at Paris Herbarium. And because I'm a systematician, what I like most, I'm kind of obsessed with trees. Many hierarchies, but although this is not a hierarchy, I like this graph view, which show you exactly the relationship between all the data that you have in your personal vault. And in particular, I said that it was possible to add tags, and I have created this tag, Khmer name, so that I know the names that are available with a Cambodian name, actually. Another thing, so I told you that I wanted to produce content in Cambodian, and actually we have few books, but we do have books written and produced by the Ministry of Environment in Cambodia, which are all in Cambodia. So this is very important. It's a guidebook on spice in Cambodia. And this is the first entry that I added, Phyllanthus acidus, which is a very small berry. Here you can see that you have the descriptions in French, in English, because I have translated the Cambodian below to English. So you can have more than one languages, and that's very nice. And when I tried to check for this plant in Paris Herbarium, I realized that there were no results at all coming from Cambodia. But what I get are specimens identified at the genus level. And if you have a closer look to these specimens, what you can see is that the label is entitled Flora of Cambodia, which was very much a surprise for me, because I didn't know that this has been done actually in Paris. And the other thing that is very interesting for me, I think, is that you have the list of the collectors, and I have he highlighted the name of Livibot, who is a Cambodian collector, and that also can link to binomia and enhance the attribution to the people, actually. So we have 185 specimens labeled Flora of Cambodia at the moment in Paris Herbarium. So what is coming next, because I'm already running out of time, but, more data to be integrated, of course, from Paris Herbarium, but from other sources as well. I would especially like to focus on the work from Dr. Suvan Moniher, who helped me a lot to start this, and she's an honorary member of the museum. She's Cambodian, and she has done a lot of work to promote plants from Cambodia. We would like to also benefit from the GeoBIF Hostel facility to produce this checklist, and maybe see if we can translate the interface and make it available in Cambodian as well. We would like to enhance the collaboration between Cambodia and France and try to reactivate. The museum has signed a memorandum of understanding with the Ministry of Environment before COVID, but we have to look into that and reactivate this. And we would like also to work closely with JBIF in Cambodia. So to conclude, what I would like to emphasize is that the open tools and the data that are freely available right now are very, I mean, I'm not a botanist, but I've been able to create a checklist, the starting of a checklist for botanical plants from Cambodia, just using those open tools and open data, which also built the digital extended specimen. And the collaboration that we would like to continue with Cambodia, the University of Phnom Penh and the JBIF node, I hope that it would help to participate into the digital repatriation of the specimens to Cambodia, which aligns with the fair and the care principle for indigenous data sovereignty. And actually those fair principles have been translated into Cambodian. So then I would like to thank Dr. Sylvain Monihur, the Herboyots team, Alan Elliott here, he's on the left, the 10 manager for Water Online, because without him, I wouldn't have been able to match the names, the plant list name to Water Online. And I will also take the chance to thank William again, William Elate on the left. He's sadly left us last year. And William, wherever you are, I hope you're happy because I finally got to get some data, Cambodian data into Water Online. And I would like to thank Nikki for her precious help into navigating into Echinopsis, and you for your attention. Thank you.
Yeah. Any questions?
Thank you very much for your presentation, and I would like to talk about flora of the Cambodia. Do you recognize how to say the project between Kyushu University in Japan to the Cambodian, how to say, collaborator? Because in the previous, maybe the previous decades, many pictorial book about flora in Cambodia, especially specific, how to say, national park, and maybe the over the 1,000-page just of the flora information is already provided in these books, or maybe it is probably, I think that it is we have a chance to collaborate your institution to this team by the Professor Yahara-san and Professor Yahara or something, Dr. Tagane or something like that, Japanese collaborator. Also, maybe the maybe they have good data to provide your project.
Yeah, that would be great. Thank you so much.
So perhaps this book is somewhat minor for not published in the major journals. Maybe many people didn't realize about that.
Yeah, thanks very much.
And it is already published in open source.
Oh, that's even better. Thank you. Yeah. Thank you very much.
Any other question? Okay, so we have the next presentation, which is going to be online. Arjun Basuroy, who was supposed to be here, could not make it, so that will be on Zoom.
Hello.
Yes,
Hello.
Arjun, we can hear you. Please go ahead.
So I'm Arjun Basuroy from Kolkata, West Bengal, India. We are presenting this. We are presenting on our work on our city park, which is a very newly born park, which is say around 11 years of age right now, and the changes that we have seen, the role of the park, what it is playing right now, what we want to showcase through this presentation. So the park is, I'm going, Vijay, can you change the slide? Yeah. So the park is only a few years old that I told. It started during 2011 onwards, and in 2013, partially it gets open to public. Eventually, we were granted 2 areas for harvesting biodiversity in a different form. So we have a butterfly area and we have a bird area that we have maintained. And in the entirety, the park is having a significant amount of native flora as well as non-native exotic flora, which are, in generally used in various parks and gardens for aesthetics and for their floral and other values. So these data is what we are showcasing is for the last couple of years data, not the entire data that we have. So this data shows that a tremendous amount of biodiversity influx that can be seen if you manage a urban park in a rightful way. In spite of the fact that there are many components which are not so eco-friendly and meant for mostly on public demand or to cater the people for having fun and frolics, but this park having a significant amount of area and a significant source of native flora has started yielding a very beautiful result. So already we have documented 395 faunal species from that area. It is except the butterfly fauna that we have. The butterfly fauna is around 100 plus right now. So it's almost 500 different faunal species that are documented from this place, and also around 125-27 species of flora that are also present in that area. Out of these 127 species of flora, there are many non-native flora that I mentioned, but most of the flora that we are propagating, that we are planning to increase, are from the native varieties that are originally found in the South Bengal landscape and in the marshland, because this area was initially it was a marshland which was converted into a city. When the city got expanded, it is converted into a cityscape, and then a part of that is this eco park. So the ideal condition that we were maintaining in that park is a very small area compared to the entire park area, but in that small area only we have mostly documented all these flora and fauna. So this whole work is for showcasing the fact that in spite of development, in spite of various other anthropogenic activity happening across the globe, it's not a local issue anymore. It's across the globe this crisis is happening. So across the globe, if we plan something inclusive, if we plan something keeping all the non-human entities in mind, then they reciprocate, they respond to those initiatives wonderfully well. So there is hope. In spite of the fact that a city like Kolkata, which is having an enormous population density, which is having a tremendous amount of people who are coming in and going out on a daily basis as well, still can have so much of biodiversity within a very congested area, surrounded by a very congested area. So as per our title of the thing, that the city oasis, yes, we can create oasis if we plan in an organized manner, if we give consideration for the non-human entities that were historically found from that area, or that the published data are available from that area, and keeping them in mind, if we can plan our thing, then yes, we can create basis. And all the data that we have gathered are into the GB platform. We have published every details of every data in the GBI platform, as well as in various scientific papers as well. So the in conclusion, what I can say, that we have to, if we are creating parks, even a small area, if we are creating that with a mindset that this will be utilized by human and non-human entities, and we keep our choices of flora, keeping them also in mind, not only for aesthetics, not only for what we like, but also those which they like, the non-human entities like, then this is wonderful. And again, if we all of us started sharing all our findings in open platform like GBIF, from where people can understand, people can get the idea of the historical data of a place, idea of the existing flora and fauna of a place, it will be tremendously helpful for planning such inclusive design. So this is what I want to tell through this presentation. Thank you.
Thank you, Arjun. Any questions for Arjun? No. Okay, so we'll move on to our next presentation, and if you have any questions, we can discuss later.
Yeah.
Okay, I'm going to present about a little study that we did with citizen science. Basically, there is a lot of data that has been posted and even currently posted, even though platforms like iNaturalist are becoming popular on social media. People like to tweet about what they see. People like to put it on Facebook. People like to make Instagram stories and so many different social networks that that are being utilized for posting these beautiful pictures that people always take of the biodiversity. A lot of these people may not be scientists, or they might not have studied science, so they don't know how to identify, but they just like to share what they see. And over the time, what we realized that even though we try to educate a lot of people to use, so to say, formal platforms like iNaturalist, still there is a lot of data being posted on other social media. And we thought we should explore to kind of get some of that data into mainstream biodiversity kind of platforms so that we can use them better. So we decided to do this with spiders. Why spiders? Spiders are pretty diverse. So just looking at India, we have about 1,800 species, and we are pretty sure there are at least 300-400 undescribed species. So a good variety of, like, a good diversity. They're pretty interesting to photograph. You see them everywhere. A typical Indian household can find about 7 to 10 species of spiders is what we have found over time, just within your house and nearby. They are very important ecologically. They are very important part of food chain, and they are also used in controlling insects. So that make them pretty interesting. So we thought, let's do it on spiders. And we have a very active and vibrant community called Spider India on Facebook. So we decided to harvest that data and look at what we can actually get out of it. So as of now, I think there are close to 9,000 members of this community. And every day there are posts coming across India about, oh, look, I saw this spider in my backyard, or I went to this forest and I saw, and people share. People have been sharing spider photographs, but what we have been doing over last, I would say, 7-8 years is educating them. Okay, it's nice you're sharing the spider. When you share a spider, you also mention the date and the location in your Facebook post, so that we can at least do something with that. And as it has almost become like a community rule for the Spider India community. And if someone doesn't post it, there will be a question within few minutes saying, oh, but you didn't mention where you saw it or when you saw it. And that's how the data has been kind of at least being, I would say 90% of the spiders, they just say, oh, I saw this spider. They have no clue what family, what genus. But then subsequently, within few hours or couple of days, there are people who would provide identification looking at pictures at whatever level. It could be just at family level, genus level, in many instances species level. So that is how this whole Facebook group has been working, and that's how we thought, okay, let's try and actually convert these into occurrence records if we can. So the workflow that we decided to follow is basically extract the data. So we developed some scripts to harvest this data into a very rough table. And then we actually had a couple of master students looking at each of these records and breaking them down into loosely what we can say Darwin core fields. So basically looking at the date and the locality and the common name, scientific name, whatever they have found. So they need to see the post, they need to see the comments and extract that data and type it in. So all these data was already harvested in a Excel file and provided to them, and all these columns were created where they will feed that in. We also taught them how to geolocate. So they did the geolocation for each of the locality. Sometimes it will be at a very gross level. They will say it was just in this state of India, which is maybe 400-500 kilometer radius, but it's better than nothing. Sometimes it will be smaller levels like county or district. Sometimes it will be a precise location. Sometimes, so a real variety of how the data was geolocated. But yeah, basically we then converted into lat long with coordinate uncertainty and all the standards, and then curated for whatever remaining fields to make sure that the state is written in the right field and districts and counties, all that thing, basically formatted in a better Darwin core format. And then another step we added was we did not want to rely totally on what was just happening on Facebook. So we involved a couple of local spider experts to quickly look at these and say, okay, whatever level can you, looking at this picture, whatever you can identify, or just make sure that whatever is written is correct, so that we don't kind of push in GBIF some really wrong data that sometimes people just write the names, the species don't, or the genus don't even exist in your country or things like that. So just to make sure that that is curated. And then we actually publish this data on GBIF. So this is the kind of workflow that we followed. So this is like a typical post that it looks, and you can see there is a little comment like this person has just said East Sikkim and like May of 2004. But we at least have a month rather than not exact date. Sometimes you can infer that looking at the post itself. It is in the month of May, it's probably the same day person has posted. So we have done some of those steps and curated the data. And as a result, we actually extracted about 20,000, it's not really records, but posts from Facebook. And after curation and filtering out, some of the posts may not have a spider picture, or some of the posts are talking of something else. So after filtering everything, we were left with about 15,000 curated records, which we could actually publish on GBIF. So this is the data set that we have published, which has already received 9 citations, 15,055 records. And yeah, so basically this is the kind of geographical and taxonomic coverage of this data set, which was quite promising in the sense that most parts of the India were covered. And I think from the places where there is not much data is probably not many Facebook users itself, or not many people who are really keen into looking at spiders. So that, and if we look at the taxonomic coverage, that also was fairly good, covering most of the families and a lot of identifications going up to genus and many times even species level. So that is how the data set coverage looks. So yeah, basically that's what I wanted to basically share, that it required quite a bit of input to curate the data, but at the end of it, 15,000 records is, I think we added almost 2 times the spider records that we already had from India at that point on GBIF. So which was a significant jump from, so a significant understanding improvement in terms of what we now know about spiders than before this data set was published. And I would like to really acknowledge the whole Spider India community members for continuously posting this, and obviously GBIF for supporting this project through their BIFA program. And thank you. Any questions?
Thank you, Vijay. I was wondering, so do you have any thoughts to why would the community rather use Facebook to post spiders and everything, and maybe if they could be encouraged to directly use some kind of citizen science platform that would already have all this in place to standardize the data and share it on bigger platform like iNaturalist or the Biodiversity India portal or something? Like, what could be done there?
Yeah, so we have been consistently doing this, and over the time, number of posts on Spider India Facebook community are decreasing, and number of posts on India Biodiversity portal and iNaturalist are increasing. But there are still some new people who absolutely have no idea what is iNaturalist, what is biodiversity data, but they want to learn about spiders, and Spider India is a kind of place, starting point for them to start joining these communities and understand. Now, a lot of times people post a picture on Facebook, but also provide a link of iNaturalist, saying, please help me identify it, or iNaturalist, rather than just providing it here. Because iNaturalist, we may not have 8,000 spider enthusiasts active from India, but Facebook has that many people. So that's one of the reasons people are still using it. And definitely there is a shift. We have been talking to people at times one-on-one, at times on phone, to make sure they understand this and start posting on iNaturalist and India Biodiversity portal.
I have two questions. One is overlapping, and one is license. First question is, how did you detect overlapping data between the iNaturalist and spider in India? Maybe some, how to say, motivated amateur scientists also posted to the Facebook and maybe to the iNaturalist. So in that case, if you're creating data from the Spider India, so maybe overlapping is happened in GBIF data. So how did you solve this problem?
I mean, in a small way, we recommend everyone to post a iNaturalist link if they have posted on iNaturalist. So those records we did not use in our curation.
I see.
But I'm very sure there is a lot of overlap which people have not, or people have identified the spiders on Spider India and later on, after a few days, gone on iNaturalist and did it. So right now we have no way to do it, but
okay,
we have tried a little bit, whatever.
Yeah.
And I always think, rather than not having data at all, data duplication is all right.
And second question is about the license. So some photographer want to keep their license. So I mean, they want to do, how to say, they do not want to become a CCBI or something, right, open license. So but of course, our GBIF is maybe it is better to publish in the open license. So I mean
So right now we are not publishing the photographs. We are not copying photographs from Facebook. We are just publishing the metadata.
I see.
We are not sharing. We are not giving link to the photograph on GBIF right now.
Okay.
But the future plan is we actually had a dialogue with few users, and the plan, we are not even identifying the users. In recorded by, we have a coded username. It's just user 150. So we are not able to identify that person right now, but we are in discussion. We had a dialogue, and we are planning to identify those users, actually attach their ORC IDs to each of the record that we have already published, and if they are willing to attach the photographs link directly, maybe Facebook link provided to the this thing, or if they are willing to take that extra step and add those pictures in, say, Zenodo repository and link it. But that is a little longer process because it needs a consensus, it needs a lot of
exactly.
social engineering to take place before they are really willing to do that.
Yes, in Japan, we have also many big occurrence data inside, how to say, Twitter or something, Facebook or something like that. But of course, that problem is still common.
I'm not sure it's the comment or question. Just like for the experience sharing, we have the citizen science project. One of my friends doing in Taiwan called Turtle Spot Taiwan, and they already like announced the license in the Facebook group. They also use the Facebook group, and if the publisher want to like publish their occurrence and photos on the Facebook group, that means they allow already about the license that they announced. So they will not have the license issues afterwards. And but another question is that, do you think the Facebook is the good place to storage the images? Like, as we know, GBIF does not store images. So if you want to like store it sustainably, what do you think to transfer it to Zenodo? But I think it would be like, it's a hard work to export all the images from Facebook.
We are playing with some scripts. We are hoping that we are able to, if not actual image storing, screenshots of the Facebook post and then post it on, for those people who are going to let us do that. We are experimenting with that. That is relatively easier to actually get a screen grab of the Facebook post and then automatically do it.
Yeah, so provide a link.
Yeah, so that is one thing we will explore. But as I said, we are still working on the social aspects. And once we have at least a kind of a group which is ready to do it within the Spider India community, then only we are going to actually start talking to more people, because I'm pretty sure there are going to be a small percentage of people who are going to also resist, saying, oh, how can you take our data without our consent or things like that. I mean, when we started this project, we have posted a message on the group saying this is what we are going to do. But I'm pretty sure 90% of the people didn't understand what we are doing. So, but in that sense, we are safe here. We have posted and we have informed them. But yeah, obviously, these are some of the questions that we will have to tackle at some point.
Yeah, I think that's the global issues for every like citizen science. Yeah, thank you.
A very interesting initiative. I'm curious to know, how do you curate your, the spiders? I mean, you mentioned initially you had 20,000 inputs, and then later on it became 15,000.
Right.
Yeah, just interested to know how you do it and how many people are involved in this curation tasks.
Yeah, so we had only 2 people working like almost full time for several months to curate this. And the drop of records is mostly because they were non-spider records. They were not real records. They were like other discussions, or they're referring to something else. People sharing, oh, a new research article has come. So these 20,000 were the posts from Facebook, out of which 15,000 were real user. Sometimes the images were really bad. And we were not even, and sometimes they were not even spiders. They were something else. I mean, strictly, we were actually restricted to that particular group, and people do post like whip spiders and camel spiders, and which are not technically spiders. These are other arthropod groups. So those records and other discussions were kind of removed. But as much as possible, we have incorporated the data, even if it sometimes, I mean, we might have few records which just says spider and it's from this particular state. which is like a 500-kilometer radius. But right now we have kept it in a hope that maybe that person sometimes revisits and does something better with that. But yeah, that is always a challenge. With identifications, we had a couple of experts quickly looking through and saying, okay, if the identification on the group was done is a jumping spider, whether that looks like a jumping spider or not, or a particular genus, whether that looks like a particular genus or not. And those are, fortunately, both of them were administrators of the Spider India group. So they also have been looking at the posts regularly and identifying and curating then and there itself in some sense when they see the post. So yeah, that's how we taken care of the taxonomic quality. Okay. So I'd like to thank all the presenters once again and all the participation, everyone who asked questions and encouraged us. And I'm sure this will open more discussions on collaborations within the community here as well as broader biodiversity informatics community. So yeah, thank you so much.